Thanks for coming to today's seminar. I think this is working, yeah. And I'm very happy to present Guillermo Monreal, who is an assistant professor in systems chemistry at the University of Strasbourg. Guillermo is from Spain. He did his undergrad in Madrid. And then he went to Groningen in the Netherlands to do a PhD in Professor Otto's lab, where he was working on self-replicating systems. I think he will correct me if I'm mistaken. And then he did a postdoc in Malin's Kamperman's lab, where he was working on spider silk mimics. So I think that's also where he started his journey about complex coacervates, liquid-liquid phase separation, and so on. So today he's here for a seminar, and I'm gonna leave the stage to Guillermo.
Okay, thank you, Mehdi, very much, and thanks everyone for coming to my presentation. Like Mehdi said, I'm assistant professor here since almost 1 year now, which is arguably not a lot of time, but I think that my team has worked very hard this year, and we got some interesting results that I am very excited to share with all of you. All the projects that I will present will be a bit diverse, but they will all be centered around liquid-liquid phase separation. So let's start by introducing that. And liquid-liquid phase separation has become quite a hot topic in the last years, and that is one of the big reasons for that. It's its connection to cellular organization. So, you know, this is a cell, and I am an organic chemist, I think, like many people here. And when we look at the cell, we compare it to something like this, because they have some similarities, right? They are both a closed space where chemical reactions happening, and those chemical reactions is what we are interested in. However, there's quite a big difference between the two, and it is that in the round bottom flask, you normally run a reaction, but in the cell, you have a lot of things going on at the same time. So trying to mimic a cell will be like me giving this round bottom flask to a student and telling them, you have to run a reaction here, and a different one here, and a different one here, a different one here, and a different one in the rest of the space. And those reactions don't have to communicate with each other, or even better, they have to communicate with each other in a controlled way. There's also spatiotemporal control. So after 5 minutes, I'm going to switch it up, and then the reactions have to move to different places. And of course, this is a completely crazy and impossible idea. If I will give it to a student, then they will be smart not to listen to me. And instead of doing the reaction in one round bottom flask, do it in a lot of tiny vials. And this is a very silly analogy to show you the importance of compartmentalization in the cell. Cells are able to do all of these impossible tasks because they have a lot of organelles and a lot of small compartments inside of these big compartments that they can use to run different reactions in parallel. And most of us know vacuoles and compartments based on bilayer membranes, but a big discovery in the last decades is that a lot of cellular processes are not modulated in those compartments, but they are modulated in what we call membraneless compartments or biomolecular condensates. These are liquid droplets with no membrane separating them from the rest of the cell interior. And they happen spontaneously due to the interactions between proteins, nucleic acids, sugars. There's many molecules that drive the formation of these. And yeah, this is really not the newest idea, but what has been detected lately is that these type of compartments are present in many functions of the cell and in many cellular processes. Still unclear in some of them what is their role exactly, but they are involved in many different cellular functions. And that has sparked a very big interest in liquid-liquid phase separation in biology and also in chemistry by extension. Now, if we look at the synthetic equivalent of this, what most people work on is coacervates. Coacervates are liquid droplets that form spontaneously in water due to the interactions between self-associating polymers or polymer. There are different interactions that can drive the formation, but the most typical one is two oppositely charged polyions. If you mix a solution of two oppositely charged polyanion and polycation, this is what you will see in the microscope. It looks like a normal emulsion, but unlike a normal emulsion, it's to me, fascinating because you have an emulsion of water in water. And the reason why this is two phases and they don't mix with each other is that inside of the coacervate, you have a big concentration of hydrophobic polymers, and that changes its properties. Of course, this behaves like an emulsion. So it starts as a series of droplets, but over time, or when you centrifuge it, then it will coalesce, and you will have two separate liquid phases, one very concentrated in polymer, one very diluted. And one of the fun things about coacervates is that they are very tunable. So like I said, the typical example is coacervates made by two polyions. And those, in those, the interactions that give rise to the coacervate are mainly electrostatics, and therefore can be manipulated quite easily by introducing salt into the system. Adding salt into it will screen the charges on the polymers, will weaken the interactions between them, and you can, with exactly the same system, simply changing the salt concentration, go from a solid to a coacervate to a fully non-interacting solution of the two polymers. And another interesting property of coacervates, why I find them cool to work with, is that they tend to sequester solutes. So, like I said before, coacervates contain quite a high concentration of these polymers. And even though they're an aqueous phase, since they have so much polymer, they're actually quite hydrophobic. And if you look at their dielectric constant, it's closer to something like DMF or DMSO than pure water. Because of that, if you take a hydrophobic molecule and you put it in this aqueous solution, it will be much more soluble inside of the coacervate than outside, and you will have that it compartmentalizes inside of the coacervates almost exclusively. And this is very cool because this is spontaneous. This is driven by solubility. So you don't have to do all the complicated steps that you have to do in order to encapsulate something into a vesicle, for example, where you have to put it into a solvent, do the vesicle, change the solvent, and make sure that it goes inside. Here, you mix it, and it almost instantly just happens by itself. Yeah, so having introduced coacervates, a bit more specifically, what are we trying to do? What we're trying to do is establish coacervates as a tool for chemists. So we have already explained how cells use this in many different processes. Now the question is, how can we, as chemists, use this? And as chemists, we already have an impressive amount of tools at our disposal, but I do think that coacervates can fill some certain applications that cannot be achieved in any other way. And a bit by professional deformation, I think that the most interesting applications are in these fields of materials, supramolecular chemistry, and systems chemistry. That's the first main goal and the main question that we are trying to answer. Of course, when you use a tool, you first need to learn how to use it. So we are also trying to establish new ways to interact with coacervates and to manipulate them in certain ways. With this in mind, I will talk today about 4 projects, or at least try to talk about the 4 of them, depends on the time. First, on the application side, we will use coacervates to recycle proteins, in particular keratin. We will also use them to control and localize self-assembly. And then on the side of developing new ways of interacting with them, we will study how they behave in presence of an electric field. And I will also comment about some strategies that we're having to achieve temporal control over coacervation. So let's start with the first one. This is a project about keratins. And maybe some of you are familiar already with keratin. This is a family of proteins that is present in wool, for example, also in hooves, also in nails, also in hair, also in antlers. So it is, I think, one of the most common proteins in mammals. It is a structural protein. So it has this hierarchical, very solid structure, which is great because it fulfills a structural function in nature. But that also means that it's very resistant, both chemically and mechanically, as it should be. And that makes it very hard to recycle. For that reason, keratin is not only in this, but it's also in landfills a lot. So per year, there are millions of tons of waste that happen as a byproduct of the food industry, and they are just discarded. And there's a big need to find new ways of recycling this keratin. The problem is that all the ways to break apart its structure also break down the keratin itself. and that denatures the protein. A couple of years ago, there was a new protocol that appeared that managed to extract keratin in relatively mild conditions. This is mild conditions for the field. It uses DTT and lithium bromide. You can probably guess what DTT is doing. It's a reducing agent. It's breaking down disulfides. Lithium bromide has a bit more of a fun function. What it does is it binds reversibly to the oxygens in the protein backbone, and then it introduces a number of charges on the keratin that makes it repel itself, and you can have a solution. But now the challenge is, how do you go from this solution back to a recycled material? And that's why we thought that coacervates could come in. This was from my postdoc in the University of Groningen, and this is work done by Jin-Woo Sun, a PhD student that I was supervising. He thought that if you have keratins with positive charges, you can just mix them with a negatively charged polyion, and that will phase separate. That was actually the case. And this phase separation is driven by electrostatics, so it's very dependent on the amount of lithium ions that you have in the system. And therefore, you can change the mechanical properties of this coacervate quite a lot by changing the concentration of lithium bromide. Now, for the people that work with coacervates before, it's a bit counterintuitive that you're having salt-induced hardening instead of salt-induced weakening of the material, but I think it's clear why. And more important than that, because this is a point that I want to make about coacervates in materials in general, the cool thing about this is that you can change the properties of this a lot simply by tuning the concentration of salt. And then you have a viscous solution of protein that you can use as a spinning dough to make materials. In this case, just as a proof of concept, we use a syringe pump and a collecting drum, and then we span fibers of it. And yeah, since this was done by electrostatics and this was happening spontaneously, we could do the whole processing at room temperature and with water as a solvent. This is very typical of coacervates and, of course, very attractive for green chemistry purposes. We published this in a sustainable chemistry journal, and they were happy with it, but the reviewers pointed out very fair enough that this involves a lot of lithium bromide, and that's not the most green and sustainable thing to have. And also, of course, it changes the properties of the material a lot that is in there. So we decided to develop a new way of doing the same thing, but being able to remove the lithium bromide after the formation of the material. For that, Jin-Woo found out, still not sure exactly if it was a reasonable, an educated guess or serendipity, that if you take this keratin and reduce its pH a lot, it will also self-coacervate. And this coacervate is a bit harder to understand. Our best guess is that when you reduce the pH, you have hydrogen bonds that want to bring the keratins together, but then you also have the charge repulsion, and the balance between these things is what keeps it in a coacervate state. You can also change its mechanical properties quite a lot, but in this case, the pH is the main factor. And salt also has an effect. If we remove the lithium bromide, it will solidify. You have different traces here because it depends on the pH as well. But in all of the cases, you end up very quickly with G prime going over G double prime, and you have solidification. And that means that we can use the coacervate to spin fibers. But if we do this in a coagulation bath, the lithium bromide will be diffused away and it will not be present in the final material. And that is quite nice because you can get it and recover it. It's also quite nice because at the end, after you have spun the fibers, the only thing that is left is keratin, and all the things that you introduced into the system to make keratin soluble, you have removed them. That means that both the secondary structure of the protein and the mechanical properties of the regenerated fibers are quite similar to those in the original wool. And that's because, again, we work in such mild conditions that we don't denature the protein at all. That was the first project. Now I will move on to something that I actually started here, which is controlling and localizing self-assembly using coacervates. I don't think I need to introduce self-assembly a lot to this crowd, so I will just show some general points and cool examples. Of course, it's the aim of self-assembly is to form complex structures from very simple monomers. You have some spectacular structures that can be formed from this. From very simple molecules, you get these double-walled nanotubes. You can also get some hierarchical assembly where first the aromatic molecules self-assemble into a rosette, and that rosette acts as a monomer. But the general point that I want to make about self-assembly is that it's all driven by weak interactions, and therefore the classical self-assembly goes to equilibrium and goes to the thermodynamic minimum. That is a characteristic of the process, and it's also a limitation, because if you want to access different polymorphs, let's say out of a single monomer, typically that's not possible. Now, of course, there are different strategies to achieve this in the years. Maybe the most trivial one is to covalently modify the monomer, because then you have a different monomer, different thermodynamics. Of course, you can go away from equilibrium, and again, that has been done a lot, and also in this city, where you basically, the fact that you have one thermodynamic minimum doesn't mean that that's the only minimum in the thermodynamic landscape, and you can use different techniques to access the other ones. But I want to talk about something much more simple, actually, which is changing the conditions in which the assembly takes place. I think it's quite intuitive that self-assembly will depend a lot on the solvent that you use. And this is maybe one of the most striking examples. You have something that does not self-assemble into any structures, just into random aggregates when it is in water. And then when you add a co-solvent, it forms helixes, but actually depend on the co-solvent that you use, you get one helix or the other one. And that only happens in a very narrow range of conditions. So it is extremely sensitive to solvent. And this is a strong example, but in principle, solvent should affect all self-assembled structures. And where I'm going with this, of course, is back to coacervates, because coacervates are liquid. They have hydrophobicity between organic solvents and water, and their properties can be changed by an external handle, in this case with salt. So the rationale behind this project, the spirit of this project, is to use coacervates as responsive solvent droplets. Let me make my first hypothesis. Like, you have any self-assembling monomer that will form a certain structure. If you put this in presence of coacervates, well, it's very likely that it will be compartmentalized inside of those coacervates by the solubility thing that we discussed before. That will already change the self-assembly, because now it's in a different solvent. Now, if you do some changes on the solution, that will affect the properties of the coacervate, and then the solvent has changed again, so that in principle could trigger yet another change in the assembly. And there you have it. You are having different polymorphs or different structures formed from the same monomer in a very simple way. And this is in principle a general strategy that you can use. Now, this is all hypothetical. I wanted to do a proof of principle to show that this could be done. And I decided to do this using a very specific coacervate that has very sharp changes as a response to stimuli. And this was the project of Malak Jaber, a master student that worked with me this year. The idea is that you take this soy glycerin that she isolated. It's a protein that is extracted from soy. The structure is not very important. If you put it with a certain amount of salt, it will self-coacervate. And this only happens in a very narrow range of conditions. If you go outside of that range of conditions, it goes from self-coacervate to hollow shells. So to imagine that, that's just like a membrane, but instead of the lipids, you have a protein precipitate. It's like a proteinosome. On the inside, you have water, and on the outside, you have water as well. Now, this is a video that she recorded where you can see the transition between coacervate and hollow shell. It's very fast, so pay attention. Immediately after she changes the salt concentration, then you get very quickly, and in the whole solution, the formation of these hollow compartments. So the idea of her project was, okay, we're going to do this strategy with these coacervates. We have supramolecular polymers in solution. If we put them inside of the coacervates, they will make something else. And then when you go from coacervate to hollow shells, you go back to the initial solvent, but now things are different. You have that these monomers were compartmentalized in a very small space, so you have high local concentration, high crowding, and that will affect self-assembly in a different way. She tried a few monomers and a few supramolecular polymers from this. They all went in a similar way. I will show you one of them, pseudoisocyanine, an organic dye. It tends to form these very large fibers, almost crystals. We picked it because it was fluorescent, and it formed micron-scale structures that we could directly see in the microscope. If you put it inside of coacervates, it compartmentalizes almost quantitatively. And you can see that this affects the self-assembly because it's not making these micron-structured fibers anymore. If you now add extra sodium chloride to make the hollow shells, that is possible, but we were a bit surprised to find that the pseudoisocyanine, in general, the monomers that we tried to put in here, will not go inside of the cavity, they will not go inside of the hollow shell, but they will be intercalated with the solid protein on the wall. And that was a bit surprising and not exactly what we wanted, because as you can see, there is no difference in self-assembly between these two. The formation of the micron scale structures is not restored. So Malak started looking into the literature and doing experiments to try to see how she could enhance the self-assembly of this polymer. And she found that actually iodide acts as an additive to make these polymers self-assemble much more easily at much lower concentrations. You go from having the monomer in solution to have the polymer completely precipitated and making fibers. So she repeated the same experiment, but this time instead of using sodium chloride as the salt, she used a bit of sodium chloride and a bit of sodium iodide. And you get a similar result where you have hollow shells and that the polymer is in the hollow shells. But if you zoom in, you see that these hollow shells have a structure now, and you have smaller, brighter spots that correspond to larger formations of this pseudoisocyanine. And as you can see, there are a few spots where there seem to be some small fibers coming from it. We have done some preliminary studies. It seems that these assemblies are the same assemblies that you have in bulk, but that's something that we need to study further and understand further. So yeah, we didn't get the compartmentalized pseudoisocyanine that we expected. Instead, we got some sort of hedgehog structure, which was a bit serendipitous, but still, I think, pretty cool. It's not the general tool that we wanted to achieve yet, because we are still relying on a stimulus that is specific to this polymer. But we do get compartmentalization, we do get spatial localization, and we do get that we can trigger the assembly. So I think it's an interesting system that is worth studying further. Moving on to the next project, we want to look at coacervates in an electric field. Why? Because coacervates are basically a ball of charges. And since all these charges are here, well, I hypothesize that if you put an electric field next to them, those charges will be affected by it. And perhaps we will get something like deformation. That's quite nice, because then we could induce forced alignment, and that could have a series of different effects on the coacervates. But before we got there, we just wanted to see what happened. And this was the project of another master student, Haidang. He followed some previously described protocol from Professor Tirel, who was here a couple of months ago. He made two types of coacervates, same composition, but different ionic strength. So one of them, you have it right after mixing two solutions of polymers, and then it has all the counterions in it. In the other one, you exchange the counterions for deionized water. So this one is a much more liquid-like coacervate with weaker interactions, while in this one, you have removed the salt and you have a much more solid-like type of particles that is in between a liquid and a cross-linked gel. If you put this in an electric field, they have radically different behaviors. So let's start by the one at low salt. This is the one that was described already. And what you see here is mainly movement. So there is an excess of charge in here, and the droplets are very rigid. And as charged particles in an electric field, they simply just move, or at least that's what was described in the paper. This is a video where Haidang was switching the polarity of the electrodes, and you can see that you can control very well the coacervate moves to one side or to the other. Now, if you do it with the weaker coacervates, well, first thing that you can see is that they spread more in the surface. And then if you put them in an electric field, what you observe is that they move, but on top of moving, they split into small particles. And that is quite interesting and quite new, because when you work with coacervates, what tends to happen is the opposite. They coalesce over time, and you end up with a film of coacervate on your glass surface, which is not ideal if you want to use them as compartments, or if you want to use them as protocells, or if you want to keep them stable over time. So the fact that we found an easy and not relying on mechanical strength way to redisperse this solution and to re-emulsify it. We thought it was pretty cool. It was also pretty hard to understand. So I think in light of time, I will not go through the whole characterization and the whole experiments that we did to try to understand exactly what are the forces governing this principle. And I will just go a bit quickly to the conclusion. The idea is that if you put coacervates in an electric field, there are different processes that can take place. You have electrophoresis. That's just the movement of a charged particle to something with the opposite charge. You also have what we call flow driven by electrochemistry. So in the conditions that we do this, you have a bit of redox reactions on both electrodes. You have a water reduction on one side and copper oxidation on the other. And that causes a net flow of hydroxide ions from one side to another. And you also have electroosmotic flow. This is a type of flow that is related to surface interactions. You have a charged surface and therefore an imbalance of charges in the solution. And that makes the whole solution to flow as well. The main difference between these forces is that one of them will affect individual particles. The other two will move the whole solution. And this I will skip a little bit. We did some investigation, mainly measuring the speed at which different particles move in different conditions. And what we found is that in this case, what you have is two opposite forces. So you have a background flow coming from electroosmosis and this hydroxide-driven flow. And you have electrophoresis that, since the coacervates are positively charged, is going on the opposite direction of this. And in this case, the more charged that the coacervates are, that's actually slowing them down and making them not move as fast. We did most of this for rigid splitting. And this is still ongoing, and this is still something that we need to characterize further. But yeah, this is at least where we are at now. In any case, regardless of whether we understand this or not, we have a new way to make coacervate split, and I think that's pretty cool. And we also observed all sorts of other interesting behaviors that we didn't even start characterizing. One of them is the splitting. We also saw that if you take a more rigid coacervate system, then you get the formation of a triple emulsion, where you have droplets of water inside of the droplet of coacervate that is inside of the water. You can see it maybe more clearly here. So this is a video where we turn the electric field off, and then those droplets are disappearing. If we went from direct current to alternating current, we also saw organization of the droplets into tiny fibers. If you increase the voltage, then you get that those fibers merge back into one big droplet. And if we do this with in the same conditions that we get splitting with the direct current, we get that there is still splitting, but now it's happening from both sides of the coacervate. Now, some of these behaviors were already reported by other people, some of them were not, but we still think that it's a system with a lot of potential, and that once that we understand the interplay between all these forces, then we can understand all of these and maybe try to use it for different things. Last project that I will briefly mention, I think that we're doing quite okay in terms of time, actually, is to establish a way to achieve a temporal control over coacervation. So if you look at these biomolecular condensates, they actually don't stay the same over time. They are not static structures. What you have is that they are all the time forming and disappearing and moving and just what we call aging. There are also some previous studies that characterize them more mechanically. And besides appearing and disappearing, you also get that over time, in this case, they become more and more solid. So this is a time age superposition, where you have that as you go from blue to red, that is older coacervates, and then they go from very liquid to more and more solid. Now, this is a very important characteristic of coacervates in the cell, of biomolecular condensates in the cell, but it has not been replicated a lot in synthetic systems. And that's what my PhD student, Lunan, is working towards. using a very simple interaction and a very simple model. The idea is that instead of using the traditional salts that you use to screen the charges in coacervates, she's using a salt that is ammonium carbonate. Normal salt, it's also a buffer, but it has the particular thing that it decomposes or it's in equilibrium with water, ammonia, and CO2. And of course, all of these are gases. So if you have it in an open system, you have that over time the salt evaporates, and therefore you have a system where the ionic strength is going down over time. And then we hypothesize that we can have a solution of polymers that remains in solution over time, and as the ammonium carbonate evaporates, then it will form coacervates, and over more time, as that evaporates, you can get the precipitation and the formation of a solid. And not only that, but through this whole process where the coacervates remain a liquid, the properties will be changing over time. Lunan screened a lot of systems. In the end, she settled down in this one, but it actually seems to be quite a general process. So there are definitely many coacervates that are affected by this. And just as a proof of concept, she just did this first experiment where you start from a fully transparent solution, keep it stirring, and then after a certain amount of time, you have that coacervates appear. Now, this was just a first experiment. Of course, if you control the initial concentration of ammonium carbonate or the stirring rate, the temperature, you can change how fast this happens. Our next step is to go from solution to coacervate to solid. And we have already seen that that can happen with ammonium carbonate. So ammonium carbonate can cause the transition from one to another. But of course, you need to go quite low in ammonium carbonate to get to the state where you have a solid. That's why we're looking now into doing this experiment at different temperatures in order to cover the whole range of salts faster. Seems quite simple, but I still think it's quite a neat way to have temporal control over the properties of coacervates. And additionally to that, and this is a very naive and perhaps optimistic way of looking at it, but I think it's pretty cool. In this system, we now have constant depletion of ammonium carbonate that is constantly being evaporated. If now you introduce a reaction that is generating more ammonium carbonates, then suddenly you have something that is creating ammonium carbonate, something that is destroying the ammonium carbonate. You can run the system out of equilibrium. And since this has an influence of coacervates, and coacervates can probably influence these processes, that introduces some nonlinearity in the system, and that at least gives it the potential to show some pretty cool behaviors like transient formation or oscillations on the ionic strength that are quite simple, are based on very simple processes, but I think it's not something that people have looked into before. With that, I would like to finish. So I want to thank my team. I think that in this picture, there is all the people that have worked in the projects of these presentations. So it's Malak, Haidang, and Lunan. And there's some more people working on related projects. Also, Jinwoo and Marlene Kamperman from the University of Groningen for the project in keratin during my postdoc. The rest of the Laboratory of Complex Systems out of Equilibrium and Professor Thomas Hermans for hosting me in the Lebel and in ICES. Thanks to all the funding sources of my students and of the group. And of course, thank you all for your attention.